Now, as I always tell you, just don't mug up things. So, can you tell me the meaning of this just from the word? Yes, because meter refers to measurement, cytometers refers to cells, hemo refers to blood here. So, blood cell counting, hemocytometer. We are using it for RBCs and WBCs, and these apparatus can also be used for some other counts, including sperm counts and all. But as it was first developed for these blood cell counts, it is called hemocytometer. Okay. So, this one will be consisting of an RBC pipette, which is called the RBC pipette, WBC pipette. You don't need to write, everything is in the books. So, this is the one, this is the box, which is together, it is written over it, hemocytometer. Okay. So, one by one, this is the RBC pipette, this is the WBC pipette, and we have to learn to identify it. This is one cover slip which is provided. Okay, cover slip. But everything is important. But what is of primary not importance, but what steals the show? Do you know meaning the stealing the show? Like in a show, who is the very significant performer? Like, I think it was, which singer came? Ankit Tiwari. Okay. If others are performing, but he is stealing the show, that means star of the show. Like that, here star of the show, this is not writing, all these are not writing, is the, obviously we can't work with the RBC, without the RBC pipette, we can't work, without the WBC pipette, we can't work. But the counting chamber or the cell is called the Improved Neubauer Chamber, and this word is important in the examination. Students are uttering Neubauer Chamber, but you have to tell what is the apparatus. Apparatus consists of RBC pipette, WBC pipette, covered slip, and will require other things also, like a petri dish for keeping the fluids and all. There are fluids which will dilute the blood. Fluids are required for diluting the blood. Those are actually, we require the experiment, in the experiment we require all those things, but the part of the hemocytometer are these things, which is provided in the hemocytometer box. And you have to call it, the chamber, you will see just now, you have to call it the Improved Neubauer Chamber. Why? When we will discuss the structure of the chamber, we will come to know why it is called the Improved Neubauer Chamber. That means, after knowing the structure, you will come to know of it, why it is called improved, but simply because there was a previous version of it and then it was improved. So, this version which we use now, it is the improved version. So, you have to call it the Improved Neubauer Chamber, not simply the Neubauer Chamber which was used before that, because things are evolving. Science is also always evolving. So, what is so except this, a petri dish required for keeping the fluids. We will show the fluids when you go to the lab, what are the diluting fluids, and we will talk about it first, that how do we perform the experiment. This is again in a small box inside the hemocytometer box. This is the Improved Neubauer Chamber. This one is just broken in the side. So, actually the counting grid for cells counting, there is a counting grid which is present in this part. One counting grid is in this part in this upper part, one identical counting grid in this lower part. There is a, see, a groove in between. So, see it, pass it on to one another. This is just broken one side, that should not be broken, that should be intact. So, many things are written, we will come to that, just see it. So, this is the, just see one groove in the middle horizontally, and there are two identical counting grids, no, not on the vertical sides, this that central vertical column, but separated by a horizontal groove. There are two counting identical grids, any one can be used, just pass it on, we will again see it. So, this is the improved Neubauer chamber as it is a version of the previous one. Some French anatomist and scientist, some Louis Malassac first discovered, or it is the first hemocytometer known to be developed in history. There was another person called Kramer. Unfortunately, the scientists have done so much for the evolvement of science, but even we don't know the full name of Neubauer. If you search on the internet also in literature, what is the full name of Neubauer, we don't know. So, that is very unfortunate. We are busy with celebrities and all, but we don't know these people who, even the names of these people who are, that means not recorded in literature, who have done so much for scientific evolution. So, see it in the horizontal, central horizontal part, but separated by a groove, vertical, but separated by a horizontal groove in the middle. Two identical chambers, one in the lower one in the upper one. Any one can be used for the experiment, we will come to that. So, just hand it back, we will again discuss about it. So, somebody named that French anatomist and scientist is known, but that is not used. Now, what is used is, even before that, that person called Kramer, some A. Kramer, he developed one in 1855. Now, then what was used was by this Louis Malassac in 1874. Ultimately, the Kramer again developed his own work in 1885, but ultimately this in the early part of the 20th century, 19 something, this person Neubauer, whose full name we don't know, is developed this one, improved the previous versions, and from then on it is called the improved Neubauer chamber, and hemocytometry is done with this thing, this apparatus. So, we just had a look, you just had a look at it. So, there are these, we talked of two identical chambers over here, and did you notice what is that? There is written, the depth is written. Depth means if we put the cover slip, it is written millimeter square, okay, forget it for now, okay, it is written in millimeter square, I will tell you how to calculate. If we put the cover slip, whatever is written, don't go for that. If we put the cover slip over here, then there will be at least a small gap, although it is not recognizable by the eye, but there will be a small gap between the cover slip and this chamber. That is taken to be 1/10 millimeter, okay, and we will require that for counting. 1/10 millimeter taken to be means that is not we are assuming, that is 1/10 approximately, this is 1/10 millimeter. So, that is taken to be that, and we will require it very much during the, for the counting of the blood cells, the gap between the cover slip. So, how we will do the experiment, we will come to that. Gap between the cover slip and the counting grid is 1/10 millimeter. Now, we are talking of the counting grid. First, let us know about the counting grid, and then we will know about the pipettes. So, just I will draw by hand, so it won't be a perfect square, I think, but just assume it to be a perfect square first, because I am not using a scale or something. So, just assume it to be a square 3 millimeter into 3 millimeter. Just assume this is 3 millimeter length, 3 millimeter breadth. So, we talked of identical counting grids. So, each one of them is 3 millimeter, a large square which is 3 millimeter in length, 3 millimeter in breadth. Okay. Now, this is divided into Now again you have to assume, I am drawing by my hand, so it won't be perfect. So these are all identical, divided into 9 squares. So what will be the length of this one? So 1 millimeter and 1 millimeter, right? Each one of these will be 1 millimeter into 1 millimeter. Okay. This one, all these things. The squares used for counting the blood cells we are talking about, erythrocytes and leukocytes, are the 4 corner ones and the central one, either the RBCs or the WBCs, but not all of them for RBCs and WBCs. So for RBC, only the central square. For WBCs, the 4 corner squares. Okay. Yes, just draw it. Although everything is in the books, I'll ask you to draw it just for, so that when you draw by yourself, it creates an impression in your brain. So 3 millimeter into 3 millimeter, you don't have to use a scale. That just draw like a square. Consider it 3 mm into 3 mm. So 9 squares, 1 mm into 1 mm. 4 corner squares are used for counting WBCs. The central one is used for counting RBCs. And why I have made a cross, those are not counting squares, those are not used for counting. Okay. So I have made a cross over there, that is, those are not used for counting. Now if you have drawn this, now again I am drawing the same thing. So now it can be seen in this diagram, but I am just making it separate one. Suppose this WBC square, this is a WBC square. So this is 1 mm and this is 1 mm. We will call these squares as from now on as WBC and RBC squares. When I am telling WBC square, it is either of the 4 corner squares. So when I am telling RBC square, it is either of the the central one. So this is again divided into 16 identical squares. Each WBC square is divided into 16 identical squares. So what will be the length and breadth of each? So take any one. Okay, right, 0.25, but I would suggest don't go for decimals. Okay, during calculation we have to do calculations for counting the cells. Decimals will confuse not only you, all of us. That means not only this is just a simple arithmetic mathematics, but multiplying something by something by something, it is always better to do it in fractions, and then at the last, if you want to convert it into the decimals, then you can convert, but that will not be required over here. Like I told, the depth between the cover slip and the counting grid is 1 by 10 millimeter. I did not say it's 0.1 millimeter. Obviously, it's 0.1 millimeter, same thing, but don't use decimals. Then it will, calculations will be difficult. So write it as, nothing to write actually, obvious, it is 1 by 4 millimeter, and this is 1 by 4, which is obviously 0.25, but say it as practice, It's saying it as 1 by 4, not 0.25. This 1 by 4 millimeter. So, each of the 16 WBC squares, like each of the either the central RBC square or the WBC squares have an area of this central RBC square or any of the WBC squares has an area of 1 millimeter square. Likewise, each of the after the division, these 16 identical squares each will have an area of 1 by 16 millimeter square. So, for counting WBCs, we have not exactly, we should not say two types, the same divided into smaller squares. So, we can say it as large WBC square and small WBC square. Large one is 1 millimeter square area, small WBC square is 1 by 16 millimeter square area. Just this will suffice. Now, we just draw the RBC square separately. So, all these, as the board is just dirty, don't get confused with the previous lines. This is the RBC square. Say this is the RBC square again. So, this is 1 millimeter square, 1 millimeter into 1 millimeter is 1 millimeter square. It is divided into identical, how many? See, I have already drawn, it should be identical. So, I have already drawn the vertical lines. So, horizontal will be the same. To make it identical, how many? 5 into 5, 25 squares. Okay. So, now whole is 1 by 5, 1 into 1. So, each one of will be 1 by 5 into 1 by 5, and do not say it as, better not to say it as 0.2, say it as 1 by 5. Okay. And at this point, I will just tell you why this is called the improved Neubauer chamber. Okay. Because earlier to this version of improved Neubauer chamber, this was divided into, like the WBC squares, this was divided into 4 into 4. Okay. Now, this is 5 into 5. So, this is the only change which has taken place in the improved version. For better counting, you will understand why, but when we talk about the counting, this is 1 by 5 into 1 by 5. Now, this 1 by 5 into 1 by 5 each square is again divided, each of these I am showing in one, but each of these is divided into 16 identical squares, each of these, not only one, I am showing only in one, just for the sake of convenience. When you will find in the books, you will find the diagrams, the whole, and I will also show you how the whole of it is done, but I am just for first time you see it as like this, just for convenience, each one will be divided into. So, if as this is a small space, if we draw this one, say 1 square from this one, if we just enlarge it, so what will be the length? It 4 into 4, the length will be 1 by 20 millimeter and the breadth will be 1 by 20 millimeter. So, now for RBC counting, again not the word should not be a type, the same square is being divided, not 3 types, but we can just name these squares as large RBC square, medium RBC square and small RBC square. In books it is being written as smallest RBC square. If we say large, medium, small, there is no reason to say smallest, but books are writing it is the smallest. Smallest is indicating this 1 by 20 into 1 by 20 square. Okay. So, large, medium, small. Large WBC square is 1 millimeter square. 1 millimeter square means 1 square millimeter area. Large RBC square is 1 square millimeter area. Small WBC square is 1 by 16 square millimeter area. Medium RBC square is 1 by 25 square millimeter area. Small RBC square is, medium RBC square 1 by 25, small RBC square is 1 by 400 square millimeter area, right. So, this is you have understood just the division of the squares, WBC square, RBC square, we have understood the divisions. So, RBC squares has 3 categories of divisions, only 1 category in the large RBC square, but 3 categories in the in case of the WBC square, but only 1 and 2 in the WBCs, and for RBCs it's 1, 2, 3. Large, medium, small. Now just let's have a look at the pipettes. You start with this one. Even the labelings have become fade. Anyway, so just by looking at this one, that is the, on the left hand side, your right hand side is the RBC pipette, and this one is the WBC pipette. Now, all of you see both, just pass it on. How to identify? What did you have a look at? So, what comes to our immediate notice is the color, RBC pipette and the WBC pipette. So, pass it on. Color of what? Color of the top of the pipette. Okay, that is colored like that, and you can see another colored thing inside that, what is called the bulb of the pipette. That central solen part of the glass is the bulb of the pipette, and that is containing a colored thing that is called a bead, B-E-A-D, nothing to write, all these are written in books. That is, yes, pass it on. You have seen that one also? Okay. So, that is what immediately comes to the notice is the red top and the red bead in case of the RBC pipette, and the white top and the white bead in case of the WBC pipette. But actually what can happen is, with time the color of the bead can fade away. Okay. So, it is not very wise to identify with the color of the bead, although the color of the top won't fade away, but even if it is, sometimes it is, that means wrong manufacture and something, but other things will help us identify. So, what immediately comes to the notice is color. So, have you seen it? Give it back. No, nothing is there. No, nothing is there actually. It is dry. Yes, that means it has been not dried properly. Okay. So, it should be dry. When we are using it, it should be dry. So, this is, now you can see it is better to identify it by other things you see. Now, can you identify the size of the bulb? The size of the bulb is more in case of RBC pipette. Okay. If you look at it side by side, you will recognize that the size of the bulb is more in case of RBC pipette, and there are labelings. The stem of the pipette is this one, inside which is the that means a part of the stem has been just made broader to make the bulb, but the stem of the pipette is this, and usually the whole thing is the stem of the pipette. Inside is the bulb is towards the upper part of the stem. Usually referring to the stem of the pipette, we mean this part. what is below the bulb. Although this is the stem, this is the stem of the pipette, but the stem of the pipette we usually refer to this part below the bulb. Okay. So, the stem of the pipette in both cases has two markings printed on it, although these markings also may fade away, 0.5 and 1, but we have to know the markings. If it fades away, so main is the size of the bulb and the markings are 0.51. In case of the WBC pipette, there is a marking 11. Okay, you can see it from here. If you take it in your hands, you can see 0.51 and 101. No need, when we will work, we will see it. So, it is here, it is written marking is 101, here it is written 1. So, what does these markings mean? These markings mean the proportion of volumes, and common mistake is taking it as like in the hemoglobin pipette there was a marking 20 and in some there are 10, then we have to repeat means we have to do the experiment twice or take it in 2 pipettes because 20 microliter is the standard volume of blood to be taken for that reaction to occur, acid hematine to form and get our experimental result. So, if it is 10 microliter pipette, then what we have to do? You have to do it twice or you have to multiply it by 2, something like this manipulation modification is to be done. Here these markings mean proportions of volume, and this word is very important. Here it is not proportion of volume or part of volume or part. So, 0.5 part of volume. It is not 0.5 microliter, it is not 0.5 milliliter, obviously it can be 0.5 liters. So, 0.5, that is common sense. So, 0.5 microliter, milliliter, nothing. It is just proportion or part of volume. This word is very important. So, whatever volume is this, one is say a unit proportion. It may be say we have never, that means thought what is actual volume is. Say this 1 is 20 microliter. So, that 0.5 is then 10 microliter. These are just marking proportions, not the actual volume by any unit. If 1 is this say roughly 30 microliter, then that 0.5 is 15 microliter. Okay. But this 1 and 0.5 are not microliter or milliliter or anything, no unit, it is just proportion of the volume, and that measurement, actual measurement how much it is not required. So, if 1 is say 10 microliter, then the above it is 11 times that, up to 1 whatever it is 11 times that. Roughly see, 1 is just below the bulb and 11 is just above the bulb. So, roughly the bulb contents, 11 will be in case of this one, the WBC pipette. So, the bulb content 10 times the volume, bulb is 10 times the volume of the lower part of the stem, or simply we call it the stem, the lower part of whatever is below the bulb, that is the stem. So, bulb, so if this is say 30 microliter, just by assumption and arbitrary figure. So, the bulb will contain 300 microliter and this is 10 times, but there is no such volume measured. It is actually 1 part of volume, bulb 10 parts of volume makes up 11 parts of volume. This 11 marking is just above the bulb. In this case, if this is just arbitrary figure, if this is 20 microliter, this is 2000 microliter, that means 2 ml, but there are no such figures. This is just 1 part of volume, this is 100 parts of volume, and so total it makes up 101 part of volume. What you may ask is, sir, this is 1 is slightly below it, 101 is slightly above it. So, roughly the bulb will be containing 100 times, just ignoring that small part. Okay. So, roughly in the stem of the pipette, that means the lower part of the stem of the WBC pipette, whatever volume is there 10 times in the bulb, and it makes up total 11 parts of volume. In case of the RBC pipette, the RBC pipette is broader, and also although don't confuse it. RBC pipette somewhat, even if we can't identify by the naked eye or it is difficult, RBC pipette is somewhat the stem is narrower, but leave that because we can't identify with the naked eye. The size of the bulb is broader and it contains 100 times the volume what is contained in the stem, that is the lower portion of the stem, or simply we call it the stem. Although the stem strictly should consider the upper portion also, we call the lower part as the stem. And remember these 0.5, 1 and 101, 111, 101, whatever figures you see are just proportion or parts of volume and does not have any unit. Okay. So, during experiment what is done is, during the experiment blood has to be sucked in the pipette as you have done for hemoglobin. So, first blood is sucked to, in case of RBC counting, in case of WBC counting, blood is sucked up to 0.5 that mark, 0.5 mark, and then fluid is sucked. Now, what happens when the fluid is sucked? Taking that pipette, fluid is sucked. When fluid is sucked, the fluid pushes the blood up into the bulb and some part of fluid remains in the stem itself. And what will be that part? That is one part of volume, because the whole blood is sucked up into the bulb. Up to which mark? We have to suck the fluid and this. When we suck the fluid, we take the fluid and this blood mixture up to this 11 mark in case of WBC and 101 mark in case of RBC. So, fluid is sucked up to that. So, this fluid is the diluting fluid. We are diluting the blood. Why should we dilute the blood? Because to count under this small counting grid, we are drawing it on the blackboard or your copy, you are drawing it on your copy, on your sheet of paper, making it large. But those are to be seen under the microscope, very small counting grid. If we don't dilute the blood, cells will be so congested that it is not possible. As you have seen, staining of the cells. So, is it possible to count the WBCs there? But is it possible to count the RBCs? No, because RBCs are huge in number. Diluting the, if we go without dilution, the cells will be congested in this small counting grid. So, we dilute the blood, so the cells are identified. Here the cells won't be identified as in staining with Leishman stain. Like RBC won't show a cytoplasm, WBC won't show the nucleus. What we will see, we will just discuss, but here the cells will be identified, WBCs will be identified by the nucleus, RBCs have no nucleus, but those structures are not of importance here. We will see cells as rounded figures in the counting grid and count them. So, talking of we will just see the fluids when we go to the lab. One is the Hems fluid for, it is after the name of somebody, Hems fluid for counting of RBCs and Turk's fluid for the counting of WBCs. So, what happens? What happens not? What does this contain? Hems fluid contains sodium chloride, sodium sulfate, mercury chloride, and mostly it is distilled water, say 100 milliliter solution. So, most of it is distilled water, and there are percentages of sodium chloride, 0.9% NaCl, and there are percentages of sodium sulfate. We should remember it, even I can't recall all the percentages at this point of time. There are for mercury chloride, but these are the contents of the Hems fluid. So, what does sodium chloride do? The role of sodium chloride to count the RBCs, we require the solution to be isotonic to that of plasma, whatever is this. So, this ensures, this sodium chloride ensures that 0.9%. Sodium sulfate prevents something called the rouleau formation. Rouleau spelling is this. Rouleau is like a French word. ROULEAUX. Rouleau formation like RBCs, if we leave blood in a, say, a test tube, so RBCs pile up, and this again is used in measurement of this principle, that this, this property of RBCs is used in measurement of ESR and PCV, something called ESR and PCV. If we have, I think we have not discussed these terms in the theoretical classes as well, we will come to know of this. So, this rule of formation, if it happens here, it won't allow us to count the RBCs. So, rule of formation, if we keep just blood in a test tube, then RBCs pile up, vertical piling up and form something called a rouleau. So, rouleau formation is to be avoided during counting of counting of RBCs in the counting grid, in the Neubauer's or improved Neubauer's counting grid. So, this rouleau formation is prevented by sodium sulfate, and mercury chloride acts as like a preservative. Okay, if the solution is this solution is such that using NaCl and Na2SO4. So, if it is kept for long, then it will not work properly. So, mercury chloride acts as a preservative, and obviously it contains water which can't be tap water or dirty water, it has to be distilled water. So, distilled water containing certain percentages of sodium chloride, sodium sulfate and mercury chloride. Turk's fluid, again water, distilled water containing two things, Gentian violet and glacial acetic acid. So, what does glacial acetic acid do? Glacial acetic acid just removes the membranes or lyses away the membranes of blood cells, not only RBCs but WBCs also. So, some books are, or one or two books are, I think, mentioning that they lyse the RBCs, but I do not think that is correct. What just mention that they lyse the membranes of the blood cells, they will lyse the membranes of the WBCs also. And then what do we count? Gentian violet does. So, glacial acetic acid does that. Gentian violet, what it does? It stains the nuclei of WBCs for the purpose of counting, nuclei of WBCs. Now, here under the Leishman stain, we observe the nuclei of WBCs as these banded ones, these in the eosinophil, the spectacle shaped nucleus, lobed ones, band neutrophils means the immature neutrophils. All these can be identified under the Leishman's or the GMSA right stain. All these we use the Leishman stain here, but here by Gentian violet, WBC's nuclei won't be stained like that. WBC's nuclei appear as roundish figures under this in the hemocytometer by staining with Gentian violet. Now, take one, what we are seeing is, if the membrane of the WBC is lysed, obviously other cytoplasmic organelles can't be seen like that, they will not be stained. So, we are noting only the nucleus, we are taking one nucleus to be one cell, one round figure. So, have you understood this? So, we are getting RBCs do not have a nucleus. So, in the counting of RBCs, if we use such a substance that will lyse away the membrane, nothing can be counted. If we can't see anything, what will we count? So, in case of this WBC Turk's fluid is such that this glacial acetic is used, the purpose is to actually remove the RBCs. If it was possible, only remove the RBCs, but as that is not possible, so membranes of all cells are being lysed, but the WBCs contain the nucleus, and that is stained by Gentian violet. Roundish figures, roundish, you can say, spots will be visible, and those will be considered each as one cell, and we will count the cells. And how we will count, we will see that. So, for the experiment what we have done, we have the Neubauer's chamber is to be set up under this counting grid is to be set up under the microscope along with the cover slip. They have to be focused for RBC counting. We have to, as well as for WBC counting, for looking at the chambers, for identifying the chambers from outside. Like we said, these squares, large square, small WBC square, or large, medium or small RBC square, we have to set it up under the microscope, and smaller these corner squares, all these are to be focused under higher powers of the microscope. For counting of the cells, in case of WBC, it's already 12:15, okay, this is actually a lengthy class. For counting the WBCs, we see the cells under this counting cells means not the blood cells, sorry, we are talking of the counting grid. This counting grid, the squares we see under the low power of the microscope, that is magnification of objective lens magnification of how much? 10. and then when we count the cells, we can count it under low power as well. This will be sufficient. WBCs are lesser in number, at least compared to RBCs. RBCs are at least 1000 more, not at least, more or less 1000 times the counting of WBCs, as we know from the theoretical value. But here what we will do, we will just count it and find it in that particular individual. The theoretically accepted value is 4.5 to 5.5 million per microliter of blood. Okay, and in the case of the RBCs, it is in case of the WBCs, it is 4,000 to 11,000 per microliter of blood. Okay. Now here, during taking in the pipette, again I am just repeating, remember these are not microliters, milliliters, anything. These are just parts of volume. When we mix the blood and the diluting fluid, what is how will we get from the result in microliter? That will be from the counting grid. We are coming to that. But the RBCs are counted under the high power of the microscope. Which is the high power? What is the magnification? No, that is the oil immersion, which we use with oil for the counting of this differential leukocyte count. Here high power microscope means 40 or 45, whatever is that will suffice. So during counting, we count it under that high power of the microscope. So that will be sufficient. So still now we have talked of the improved Neubauer chamber. What is the structure of the counting grid? What are the points of identification for the RBC and WBC pipette? And the most important point for, if we consider the two pipettes side by side, is the size of the bulb. Size of the bulb is obviously more in case of the RBC pipette. Markings are important, but markings will fade away. But it is better not to go by the color, because at least if the manufacturer is wrong, or if the color fades away, okay, color of the bead fades away, then it may confuse us. So we should not go by the color, we should go by the markings. 10111, even if the marking fades away, size of the bulb will help us identify the pipettes, which is the RBC one, which is the WBC one. Obviously, if we go to count RBCs with the help of the WBC pipette, then again it will be erroneous. So for RBC counting, we need that much more dilution. Now how much times the blood is diluted, we will come to that. For that, first we need to know what is done in the experiment. When this improved Neubauer chamber along with the cover slip is placed under the microscope, after taking the blood and the diluting fluid, we are to mix it. Okay, just mix it like this, just simple with the hand. Mechanically we just mix it so that the blood and the diluting fluid get mixed up. Where it is getting mixed up? Inside the bulb, and maybe in the part of the small part of the stem above the bulb, but usually we refer to the stem as the lower part. No blood and fluid is getting mixed up in the lower part, this stem part. Why? Because the fluid has already pushed up the blood in the bulb. Okay. So in this part there is only diluting fluid. In case of the RBC, it is the Hayem fluid. In case of WBC, it will be the Turk's fluid. There is no blood in this part. Okay. Whatever blood and diluting fluid blood is getting diluted is in the bulb, and the small part of the stem above the bulb in that part. So if we refer to the markings, above 1 and below 11 or 1 to 11, here is blood and diluting fluid getting mixed up. But no, that is the blood is being diluted. Mixed up, the word mixed up means, the word mixed up is not written in books and we should not use it. That is, the blood is getting diluted. Blood gets diluted between 1 and 101 in case of RBC pipette, or from 1 to 101 and from 1 to 11 in case of WBC pipette. Now this blood, this diluted blood is to be charged in the Neubauer chamber. The word used is charged. Charged in the Neubauer chamber, how? In the small gap between the cover slip, and this has to be gained by practice. You have to do it, charging it without forming air bubbles. okay, and so that it is uniformly spread in the upper counting grid and the lower counting grid, so that we can use any of the counting grids. If it is not uniformly spread, then again the results will be erroneous. So that blood is uniformly spread without any air bubbles within the, we will do that in the lab. So we have to charge that count with the pipette inside the counting chamber. Now before charging, what we have to do is just discard, just by blowing out we have to discard this part, okay. So this part does not contain any blood and will not contain any, obviously not contain any blood cells. So we have to discard that part, okay. Then only, and that can be seen. So while discarding, only this part will be discarded. So what will be charged is containing diluted blood. Diluted blood, blood and diluting fluid. That is the word I am using is mixed up. It should be called diluted blood, not this part where there is only fluid. This fluid has pushed up, so that part is to be discarded. So out of 100, if it is in case of RBCs, if it is 101 parts of volume total, then one part of volume is discarded. In case of WBC, if 11 parts of volume of this whole thing, if it is 11, then one part is not containing blood, that has to be discarded. So what remains is, what is the blood? Diluted blood is 100 parts of volume. The, in case of RBC counting procedure, the diluted blood is 100 parts of volume. In case of WBC is, it is 10 parts of volume. Whatever volume of blood is taken, 100 times of that is diluted. That means diluted blood is 100 times of volume of the original blood. Is it? 100 is the whole thing after dilution. Original is how much? Original was, blood was 0.5. 0.5 part of volume is blood, 100 parts is the total blood along with the diluting fluid, diluted blood. So dilution of blood is actually 200 times in case of RBC. Do you get it? If it is 0.5 part of volume, it has been diluted to 100 parts of volume. So the dilution, this is called the dilution factor. This is called the dilution factor. The blood has been diluted 200 times. In case of WBC, the blood has been diluted 20 times. So more number of cells, more dilution is required. If you take by mistake blood up to 1 instead, by mistake or even if intentionally blood up to 1, then the dilution will be 100 times or 10 times in case of WBC. But we should not take it. Why? Because this has been experimentally seen that this dilution will be favorable for counting the cells. If we take blood up to 1, then obviously again the cells will not be as sparse, because we are to count the cells in a small grid. If the cells are huge in number, if it appears huge blood has not been diluted to a certain extent, to an optimum extent, then cell counting may become difficult. That is why we have to take blood up to 0.5. But mathematically speaking, if we take blood up to 1, we will just consider that we have diluted the blood 100 times. During the final calculation, we have to use that dilution factor. That blood has been diluted so many times. If it has undiluted blood, then what would be the count? Okay. So if we dilute it 50 times, then also mathematically the result should come. Okay, but it has been seen that diluting it 100 times for RBC counting, 200 times for RBC counting and 20 times for WBC counting is just optimum to count the cells without any difficulty. That means easily or at least conveniently. But mathematically, if we take blood up to 1, the dilution factor will be less. That means the blood is less diluted, but counting may become difficult. Okay. because RBCs are so huge in number, especially RBCs. The counting grid is made such that, so that this dilution factor will be sufficient. That means first the counting grid was made, then it was seen how the dilution factor works, what dilution factors works best for this counting. Now, charging of blood, we have to learn in the lab how to charge in the blood with actual blood. After charging of blood, the blood is uniformly distributed in the counting grid. Now, how do we count the cells? How do we identify and count the cells? That is one part of this discussion. So, okay, we have to tell one or two things about that also. That suppose this is the, what I missed out is not missed out actually. If we talk of this whole square, this is a very simple thing. Just look at this. Here, the non-counting grid, in the non-counting squares, they contain triple lines. Okay, we'll show these triple lines. The pictures are there in the books, and we can, when we see under the microscope, these triple lines will be visible. These triple lines will actually go through all these squares. These small squares, all these squares means except these squares which are these non-counting squares, also through the RBC squares, and those triple lines will itself divide these RBC squares into the smallest RBC square from the medium RBC square. We can just note it when we note the, I am not drawing it here, it will look clumsy. So, this is 5 into 5 like this, and these triple lines will divide it into that 4 into 4 for each medium sized RBC square. Now, just consider where are to count the RBCs. WBC counting is more or less simple. Even we do not require the small WBC squares for identifying the cells separately. What happens is that this is a large RBC square for the smaller purpose. When we actually see it under the microscope, you can find it. When you count the cells, cells are lying over here. Okay. And cells will be also lying not only inside the small squares. If we consider it a large square, the cells will be lying in the squares and also on the margins of the squares. So, squares are made by us human beings. So, squares are the cells, when they are dispersed uniformly, they would lie on the margins as well. So, what is followed is that when you count cells, these are small squares. When you count the margins, in case of WBC squares, the square looks pretty large, so that they are counting is not that difficult. So, cells will lie all over this place. So, even if they are over the margins and you count like this 1, 2, 3, 4, 5, 6 like, okay, these margins can be easily identified. Okay, and you can count the cells as a whole. We can count the cells as a whole and say count, given just an arbitrary figure, say it's 30, it's 40, something like this. We count 40 cells in this whole square. Whole means large WBC square means what is this? What is this length? 1 mm. This is 1 mm. Okay. So, we can count the cells as a whole. But if there is confusion, to avoid confusion, at least for the beginners, it is better that you count using something called the rule of L. Rule of L means when you count this, if don't go for the large square as a whole, count each separately. Okay, so this contains, so if it is say 2 here, 3 here, say again 2 here, 5 here, like this, and then add up those numbers. So, when you are counting, what happens? So that no cells are counted twice. So, you take this, when you are counting this square, take the left and the lower margin and don't take the right and the upper margin, ignore that. Even if cells are lying on that, when you say you are counting the upper left square of the WBC square, upper left small square of the large WBC square. So, when you are counting this, count this margin and this margin left and lower and leave out these two. Then when you are counting this next square, take this left and lower, but don't count this. then this one, this one, this one, this one. Obviously inside you will count. Inside we will count, but for the margins we will count only the left and lower, left and lower, left and lower. So just think of it, if we go on like this and complete up to this, all the cells have been counted, the area inside has been counted and this margin has been counted. At last you count only this one, okay. So have you got it? So count the left and lower margin including the space inside obviously. So after counting all 16, this area is covered and this left and lower margins of the large square is also covered. What is left is the right and upper margin of the large square, that is counted at last. Even if these are a very few cells, if this is even ignored, the count will not, that means very much, but I think we should count it because we should not, with approximation as much as possible. If it is possible to count, then obviously count the right and upper of the large square at last. Then you will get the whole number of cells, okay. Similarly, in case of the RBC squares, but RBC squares is a bit complicated than this. If you understand step by step, it won't be that difficult. In case of the RBC squares, if we take 1 millimeter into 1 millimeter RBC square is this, then it will contain how many? 25 squares. So out of these 25, if we go try to count, RBCs are such huge in number, if we go try to count all these 25 squares, firstly it is not possible to count as a whole. RBCs are huge in number. It is even not possible in a short time to count even all the 25 squares. What is done is, counting is done in this, and here it is labeled. We label it as, we just label because so that we can calculate later. Left upper, this is right upper, this is right lower, left lower, and central. Central is this, okay. Most central of the RBC square is this. So these 5 are counted and counting is proceeded. We will see how counting proceeds. So these 5 are counted, not all the 25 squares, right. Now if we draw this, any one of these again separately, just for understanding, what will be the division? Say we just draw the left upper one, all of them will be identical. How many divisions? 16 small RBC squares. If we say large, medium, small, no need to say smallest, it is small RBC squares. So now these small RBC squares, 16 of them will be counted according to the rule of L, okay. RBCs are huge in number, count all these, the inside space for each of these 16, take the rule of L, left and lower, ignore the right and upper, and at last count the right and upper margins to get what? To get the count in this cell only. In case of the right upper square, count this one and get the counting of this cell only. See, count the small 16 squares, using the rule of L, count the right and the upper border at the last, and you get the square of left upper, you get the count of right upper, you get the count of right lower, left lower, and the central one, okay. Now how to identify, but before that comes, how will you identify which is the left upper square? So again the grid looks like this, grid is like this. So it is divided into 5 into 5. So on that 5 into 5, which is the left upper square? So that we will see under the microscope. See, to the left of the left upper square, it is a non-counting square. Above the left upper square is a non-counting square, and non-counting squares does not contain squares like WBCs and RBCs. only contains triple lines, which we talked about but not seen till now. Diagrams are given in books and we will see when we see under the microscope. So, left upper square can be identified by presence of triple lines left to that and above that. Right upper square can be identified by presence of triple lines right to that and above that. Right lower square can be identified by presence of triple lines right to that and below that. Left lower, left and below will be triple lines. So, we go on using the coarse adjustment screw of the microscope. So, it is very important to know the parts of the microscope, how to adjust the microscope, not only for moving it up and down, we have to move from side to side. We have to identify what is left and right of the squares in this case. And how do we do that? First we can do any one of these, we can count. There is no, actually there is no order, there is no specific order that we should count, but conventionally what it is done, first is counted the left upper one. You can do it anyway. You can do it first, you can do the right lower one, first the left lower one, then proceed to the other ones. But conventionally, left upper one is identified how? Left there will be triple lines, above there will be triple lines, and on the other sides there will be identical squares. This and this and this and this all will look same. So, on the other sides, that means the right side and the lower side of the left upper are similar or identical squares to that one. But on the left and the upper side are triple lines. So, identify the left upper square first, then what we do is by using the coarse adjustment screw, when we have counted this, move 1, 2, 3 and come to this. Okay. It will automatically come to the right upper square, but just confirm it, there will be right triple lines and above there will be triple lines. Once we have identified any one of these corner squares, others are easy. Just move 3 squares and go there. Just go below 3 squares, again using the coarse adjustment screw, go there. After counting this, just again move left and go 3 squares, and this can be confirmed by the triple lines in the, that means in the whatever direction it is for which square, the triple lines confirm it, but just moving, once we have confirmed one corner square, then others are easy to find, just moving 3, moving 3, moving 3 in the required direction. How to identify the central square? Once we can count any square anywhere, we can count any square in any order, we can start with the right lower, but conventionally we start with the left upper, we go this is anti-clockwise or anti-clockwise? This is clockwise. So, clockwise we go, after reaching the left lower, what we do is move 2 up and 2 to the right and get central. If we say, if we want to start from the right lower, then complete up to the right upper, then come down and go left to reach the central square. So, central square can be identified by central square. In case of the central square, there will be identical squares all around it. So, central square not difficult, just after the last square go up and then go to, if you move to the central, you have counted all these, move to the central from the lower left, then go up to and then towards the right to. But if you have say started from here, then complete left upper, right lower, then go. But I will ask you as beginners at least, follow what is conventional, start with the left upper. Just identify triple lines left and above the left upper, and then you move 3 up with 3 squares, whatever 3, that means 3 actually 1, 2, 3, 4, whatever is in the required direction in this order, left upper, right lower and left lower and central. So, how to count the cells, that can be shown here, that has to be shown under the microscope. We are counting, that means we have already discussed the theoretical part of it, rule of L, but actually how the cells look like, we will see it in the lab, not today but next day, on Monday we will see. So, left upper, right upper, these are counted. So, after that counting of cells is done in both case of RBCs. Here the counting, the final our figure, which the total RBC count or the WBC count as it is has to be obtained. So, we have known of the areas of all types of square, again we should not say type, it is the same square which is been divided, that means all sizes of squares we have classified as those areas we have known. Why is that 1 by 10 millimeter, the gap between the cover slip and the counting grid important? that will give us not actually the volume of the square, because square is itself, the counting grid is itself already, that means it is already made, that is not actually a three-dimensional thing. But what happens is that if blood is uniformly spread, and these things are not so clearly written in the books as I have read them, blood is uniformly spread, so it is the volume of the blood is which is being considered, but very roughly we say the volume of the square, because volume of the squares have already been made, and this is not written what I am telling now in books like that, but just think of it, volume of square we can't consider, squares are given in the grid and they are more or less like a two-dimensional because there is no depth. When we can consider volume, we can consider the area of this floor, we can consider the area of this roof, but unless there is some space between the floor and the roof, we cannot consider any volume. For the floor, we have to say area, we can't say volume of the floor, volume of the roof, we have to say volume of the room, because there is a space between the floor and the roof. Until and unless the cover slip is kept, there is no meaning of the word volume here. So between the cover slip and the squares, what it is? Blood. So blood volume we have to calculate, and the area of the squares will be needed for it, that blood is covering that particular area, and how the blood volume comes from that, knowing the height or this gap between the cover slip and the squares, and this is not explained like this in the books. The books are writing volume of the squares, but just think of it, strictly speaking, volume of the squares mean nothing, volume of the floor means nothing. There has to be a space between the floor and the roof, so you can say the area of the roof, the area of the floor, but the volume of the room, like that. If it is a square room, then it is like this, 1 millimeter, but rooms, speaking of rooms, it usually the length and breadth differ, it is not a square, it is like a rectangle. So volume of the blood in, say, we first talk of WBC, it is simpler. In one single large WBC square, what is the volume of the blood? 1 into 1 is the area of the square, so the blood is uniformly distributed, so that blood, now volume, the depth, this is called the between the cover slip and this is called as the depth, so that depth is 1 by 10, so the volume of the blood content just over 1 WBC square. In books it is written content in the square. In the square it is meaning just over the square. Blood is going between the cover slip and the squares, between the counting grid. So volume of blood there is simple, millimeter cube. So we count 4 WBC squares. So the volume of the 4 blood in, or actually strictly speaking it should be just overlying the 4 WBC squares is this thing, and this comes to 2 by 5 millimeter cube. Okay. Now again, don't go for 2 by 5 is actually 2 by 10 is 0.2, so 2 by 5 is 0.4. Don't write it as 0.4. Until the end you carry on as fractions, don't go for decimals, that is my suggestion. Decimals, calculation mistakes may happen, calculators are not allowed, so you have to calculate it in your own mind, and these will be simple calculations, so you have to bring the final result. There will be certain instances when figures will be given in questions, some mathematical problems will be there all throughout this, not only physiology, but all throughout this, like something called RBC indices will be discussed in the respiratory system. You have to calculate some things, pulmonary arterial pressure and all these things. Some figures will be there which will not be possible to calculate mentally. So what we follow is that if you show the logic, if the candidate can show the logic, full marks is awarded for subjective questions. For MCQs, usually the figures are given, the final figure is given in the answer. So usually figures are given such that the calculation is mentally possible, because mobiles, calculators, these are not allowed during the exams. But this is pretty simple calculation, how I will show you. So 2 by 5 millimeter cube of blood is what we are considering. Now say we have counted this number of cells we have counted. First we consider, just we take that figure as say n number of cells. Whatever we have counted, that is say n. So 2 by 5 millimeter cube of that blood contains how many cells? Say WBCs, n WBCs. So 1 millimeter cube contains n divided by 2 by 5, that comes to 5n by 2 WBCs. Now just if we put n, then we will get the figure, but again remember, just recall, not remember, just recall that this is diluted blood. Okay. So diluted blood will contain 5n by 2 number of WBCs, right? Because the counting area is 1 into 1 millimeter square into 4, but the volume of the blood, don't say the volume of the squares, although it is written in books. Volume of the squares is not a very appropriate term here. It should be volume of the blood. So volume of the blood overlying those squares is this. So 2 by 5 millimeter cube of blood, but what blood is this? Diluted blood contains n number of cells. So 2 by 1 millimeter cube of diluted blood contains 5n by 2 number of cells, that is n divided by 2 by 5. So 5n by 2. So 1 millimeter, and here you have to write, I am not writing, don't just give an arrow. So 2 by 5 millimeter cube of diluted blood contains n cells, 1 millimeter cube of diluted blood contains 5n by 2 number of cells. So 1 millimeter cube of, sorry, diluted, yes, diluted. So undiluted blood contains 5n by 2 into the dilution factor, that is 20 in case of WBCs, it will come to 50n number of cells. Just put n over here and get your answer. So if n is say 100, then it comes to say 5000. If n is say 150, it comes to 7500, like this, okay. So don't say volume of squares, obviously say area of square, but volume of blood overlying the square, because blood is uniformly, the reason for that is I have charged the blood in a proper method and the blood is uniformly distributed all over, yes, just overlying the counting grid, and any one of these upper or lower counting grid can be used, no need to use both. If we can use both and take an average, because humanly we are making these human errors are possible, not only errors that although this is uniform distribution we are telling, some non-uniformity will be there, and obviously in the upper grid and lower grid actually some variation in count will be there. So if there is time, it is better we have two grids in the same counting chamber, we first consider the upper grid, then consider the lower grid and take an average. This is better, but usually conventionally that is not done. Any one grid is used and the cells are counted and the answer, that means the result is obtained, okay. And only counting from the examination point of view, counting one grid and completing in 45 minutes, it will be a challenge, okay. So we have to practice it. So we won't take 1 or 2 classes for it, maybe 3 or 4 classes for this, at least 2 classes for RBCs, 2 for WBCs, like that. Except this one is just for letting you know about the chambers. So now the counting of the RBCs, again like as the squares are just diverse, so not very complicated, you have understood as you have understood the whole thing, it will not be complicated anymore, the calculation part. So how many, just to draw again over here, 1, 2, 3, 4, 1, 2, 3, 4. So 5 into 5, and we have counted the left upper, right lower, left lower and central, okay. Total count is right as n like before. If it is n, total count is n, first we consider n, we put the value of n at the last. So left upper, what is the area of the square? 1 by 5 into 1 by 5 square millimeter. So the volume of the square, not the volume of the square, the volume of the blood overlying the square is this much cubic millimeters. Okay. So left upper, right lower, left lower, central, considering all these 5, it will be into 5. So this much cubic millimeters. This comes to 1 by 50. Now 1 by 50 millimeter cube or cubic millimeters of diluted blood contains n number of cells. Right, you have got this. So 1 by 50 millimeter cube contains n number of cells. Yes, 1 by 10 millimeter. I have repeated it twice or thrice. 1 by 10 1 by 10 is the gap between this covered slip and this, and this is the whole concept. The blood is overlying in that part. That was what I was trying to explain. That is the space. This height has to be considered during taking the volume of the room. It can be the volume of the floor. It has to be the volume of the room. So that space is a very small space, but that gives me the volume of the blood overlying the squares. The squares itself doesn't have any volume. That is like a 2-dimensional. So that 1 by 10 comes from there. So 1 by 5 into 1 by 10, this is the volume of the blood overlying 1 medium RBC square. So we have counted 5 medium RBC squares. So the volume is 1 by 50 millimeter cube, which contains diluted blood, n number of cells. So 1 millimeter cube of diluted blood contains 50 n number of cells, n by 1 by 50. To convert into undiluted blood, we have to multiply the dilution factor, that is 10,010. Now just putting n, if n is around say 10,000, n will be actually, so if a, whatever is n, whatever you calculate, you are calculating, whatever is n, just multiply it by 10,000, okay. So if it is say 200, then it will come to around something. If it is say 400, it will come to around say 4. So if n is say 400, so it comes to 1, 2, 3, 4, 5, 6, 4 million, okay, 4 million per millimeter cube of blood, okay, per millimeter cube of blood. So RBCs will be around that. So, but I will not encourage that on the first day. So just know theoretically, if the value has to be correct, then it has to be come around, it has to be 5 million. So n should be around 500. So in each medium square, if n has to be 500, if in each medium square you are showing more or less 100. Now see, don't actually count cells and then write. Although we are targeting uniform distribution, it cannot be wholly uniform. That means it cannot be that 100 cells in each medium RBC square. So maybe 90 in some and 120 in another, you count actually, and the figure should come around 450, 550, 500, maybe 600. So if it is 600, the count is coming to 6 million per millimeter cube of blood. But don't show 100 in each one. So the examiner will just consider that you have not counted and you are just writing out as 100. Okay. In case of the WBCs, whatever, it will come as what we showed 50N. So 50N to make it 5,000, say if it is 10,000, WBCs actually theoretically vary between 4,000, 5,000 to 11,000, 12,000. We theoretically speak it as 4,000 to 11,000. So WBC should be like that, but actually count and write, don't count, write the same number of cells in all the 4 squares. Okay. You count and write. So it will vary at least some. If it is say 5,000, so it should be say 30 cells in one large WBC square, 25 in another, maybe 40 in another. Okay. Like this you do it. So this is okay. Just one more thing, that theoretically we accept it as millimeter cube and microliter per millimeter cube of blood or per microliter of blood. I had explained once in the explain means this is nothing much to explain here, that in the SI system whatever is microliter, that is in the CGS system millimeter cube, we can derive that from 1 liter and 1 decimeter cube are equal in both the systems. So 1 liter if we go from millimeter cube, 1 millimeter cube is 1 millimeter, write it as 1 millimeter whole cube, right? So 1 mm is 1 by 100. This will be it. I have not memorized. I am just 1 millimeter cube is 1 by 100, or better write it as, this case better write it as 10 to the power minus 2. So it's 10 to the power minus 6 decimeter cube, which is 10 to the power minus 6 liter, because decimeter cube and liter are same, and this is 1 microliter. So expressing it in millimeter cube is identical to expressing it in microliter, just that the counting systems are different. So this is more or less, I think I have whatever theoretical discussion is to be made on this has been made. We'll just go.
RBC count করতে যে বক্সগুলো আমাদের প্রয়োজন, যেগুলো আমরা দেখবো RBC কাউন্টের জন্য, সেই জায়গাটাই ফোকাস করা হচ্ছে প্রত্যেকটাকে। ভালোভাবে দেখবে স্যার, যে বক্সগুলো বলেছে দেখা যাচ্ছে, বুঝতে না পারলে বলো। এই ফোকাসটা করে রেখে যেমন এক্সাম টাইম নিচেকে ফোকাস করতে হবে।
Which has been focused here? Total central. So the total RBC square, large RBC square.
আগেরটাতে দেখলাম, আমি ওটাতে যাচ্ছি। পুরো বক্সটা রয়েছে, পুরো নিউবার চেম্বার দেখেছি ফার্স্টে। আমি এখন জাস্ট আরবিসি যে কাউন্টার বানার যে বক্সগুলো প্রয়োজন, আমি মিডিল পোরশনে ফোকাস করে দিচ্ছি। এরপর ফোকাস করবো যেগুলো আমাদের ডাব্লিউবিসি কাউন্টের জন্য প্রয়োজন, যেমন হচ্ছে আপার পোরশন, ডাউন পোরশন, যেগুলো প্রয়োজন সেগুলো প্রত্যেককে ফোকাস করবে, প্রত্যেককে দেখাচ্ছি। দেখা যাচ্ছে রাইট আপার, তারপর ঠিকভাবে দেখো 5টা করে বক্স রয়েছে, ঠিক আছে? একটা বক্সের মধ্যে আরো 16টা করে বক্স রয়েছে। ভালোভাবে দেখো, ফোকাস রয়েছে।
Only RBC, central RBC square. So you have to play this in your mind again and again.
এখানে কিন্তু WBC এর কোনো বক্স নেই, যার আছে সবকিছু কিন্তু RBC কাউন্টেড বক্স রয়েছে।
আর কততে ফোকাস করছো তাহলে বলে দাও, তাহলে see 10x. So in the 10x you are 10x objective length. What is the total magnification? 100, because the eyepiece length is also 10.
রাইট আপার পোরশন যেটা রয়েছে সেটা ফোকাস করেছে। রাইট আপার বলতে দেখো আমি একটা বোঝাচ্ছি, নিউবার চেম্বার এই সাইডটা হচ্ছে রাইট আপার, মিডলে আরবিসি, এটা হচ্ছে লেফট আপার, এটা হচ্ছে রাইট আপার এখানে ফোকাস করেছে।
So just look towards the board ones, this screen ones. She is telling what she is telling. This is the right upper. Okay. Now, what we spoke of right upper in the class, we are speaking of this one, not the right upper of the RBC. Right upper of the total Neubauer chamber. That is 1 WBC square. Just look at that.
বোঝার জন্য দেখো লেফট সাইডে